Mr. Pu Xue Mason is a graduate student researcher focusing on microbial cell factories, which have been extensively engineered to produce chemicals, fuels, and materials. He completed a bachelor's degree in chemical engineering from Rensselaer Polytechnic Institute and is currently pursuing his PhD in chemical and biomolecular engineering at the University of Illinois Urbana-Champaign. Mr. Xue's research recognizes that the metabolic engineering of microbial cell factories remain time-consuming and labor-intensive. And a major reason is the lack of a general platform for large-scale mutagenesis and high-throughput screening. And to address these limitations, he has performed genetic engineering and phenotypic screening by integrating the design, build, test, learn cycle with a biofoundry. And it is our pleasure to have him share his findings with us here today. Mr. Xue, please.
Hello, everyone. My name is Pu Xue Mason from University of Illinois. So in one sentence, I work with Robotic to solve bioengineering questions. I got the inspiration from undergraduate chemical engineering unit operation lab, where each equipment plays its own role and when integrated, fills the ultimate goal. I applied the similar concepts and used the robotic arm to connect each equipment and build a fully automated platform, which can help researchers perform daily repetitive experiments. As a bonus, they can focus on more complicated design tasks. So the scope of iBioFAB is to develop automated processes for biological researchers, include metabolic, protein, and cellular engineering. Moreover, it can do some basic cloning work and the DNA assemblies. And then in the future, we want to apply the robot system to help us to do screening of desired phenotypes. And here I listed the available equipments and the modules and its corresponding workflows in the bottom left side. And all this can be connected to each other to perform the multiplexing tasks. And all this has been connected using the Momentum software, where the scripts are behind and helps the robot to do the physical liquid handling and do analysis in the downstream end. So what we want to do is by using the automation to do a more rapid design, build, test, learn cycle to fulfill the synthetic biology goal. And overall, we have developed this DBTL cycle and perform tasks using the iBioFAB robot to develop a computational design framework and to build the processes handling cells, cell cultures, and even strain validations through our test module and the build module. And in the future, we also want to integrate the learn module to our DBTL cycle, where the robot can help itself to determine what's the next task or next step to do after running several rounds of mutations of the libraries. And in one project, we were working on making plasmids in a large scale to make thousands of plasmids that can be used in synthetic biology. So as scientists in biotechnology, this audience may already have the knowledge that the plasmids are very important in our daily life. And now I just want to give emphasis of why we want to study plasmids. So the construction of plasmid DNA from smaller DNA parts to form complex DNA systems, such as metabolic pathways, is important for the DBTL cycle in synthetic biology. And the plasmids can be used for delivery of important materials into cells or engineering of various pathways. And some genes can use plasmids as a vehicle to enter the cells for downstream processes, as I shown in this figure. But now the difficulty is that the assembly of these complex plasmid DNAs often requires trials of multiple techniques. So in the end, we can conclude that the construction of plasmids is still time-consuming and labor-intensive. So we want to find better ways to build a lot of plasmids and help them to express in certain cells. So that comes to our strategy used in this project, where we applied the agar-based artificial enzymes to do the cutting and the ligation. This was done manually before, and what we want to do is to fully automate this part. So some background knowledge, the PFAGO uses the phosphorylated 16 nucleotides single-stranded DNA guides to cut a single strand of DNA and cleaves the ssDNA target between the 10 and the 12 nucleotide position of its ssDNA guides. And these two precisely designed guides enable cutting of the double-strand DNA with sticky ends of any desired size. So as you can imagine, we can cut the DNA fragments at any locations and then reassemble them to form new plasmids. And this method has been demonstrated successful to assemble up to 18 kilobits of plasmids from 11 fragments in a one-pot reaction. So what we want to do here is to integrate our design, build, test cycle into this artificial enzyme-based method by using our iBioFAB system to create an automated workflow for construction of multiple plasmids at one time. So there are four major steps. The first is we want to build a user-friendly web front interface to help us to design the oligos. And the second and third step is to prepare the place for PCR reactions and perform the one-pot digestion, ligation, and the transformation. And in the end, we can confirm our generated plasmids using restriction enzymes and the sequencing. So in details, we want to build this iBioFAB as a service to our own campus facility or even to other universities as a collaborator to do the made-to-order plasmids. So to do that, we already built a web front interface where researchers or customers can design their singular or libraries of plasmids and search our inventories to generate their own plasmids. And once that happened, our iBioFAB technician will take the orders from the customers and place the orders through the other companies to help us to generate these primers. Then the robot can do the one-pot plasmid assembly, starts with PCR reactions in a high-throughput manner. So here I showed the detailed three modules of our robotic plasmid assembly module with fragment preparation through PCR, the actual assembly, and the confirmation step. In each of these steps, I listed the input that we need and the output of each module. And you can see I want to emphasize here is the human intervention is very low. With our researchers only need to confirm the correct amplification or correct colonies being generated. If not, then we just repeat the modules using the robot. So by doing this, we can build around 100 plasmids within 3 days using this iBioFAB system. And in the future, we plan to, as I said before, make this as a service to the campus. So we called the website as a plasmid maker, where the customers or researchers can build their own plasmids. And we are currently also build up our LIMS system, which is the laboratory system that can help us to build a lot of inventories with a lot of templates of plasmids that the customers can just select from our database. And in the end, after the robot we can generate the correct plasmids and send back to the customers. So in the end, I want to give my acknowledgements to my professor advisor, Professor Huimin Zhao, and all Zhao group members. And also I want to thank my parents a lot for their support. And here is just a short story that I have been diagnosed with type 1 diabetes during my PhD, but I got the support from my Zhao family and as well as my parents. So they helped me to got over that difficult times. So I really thank the opportunity to study in Huimin Zhao's lab. And I also want to thank the audience for your listening today. Thank you very much. And I will take any questions from here. Thank you.
Right. Thank you very much, Mason, for the very interesting talk, as well as sharing your personal experiences in your research journey. So we have some questions coming in from the audience. And the first question is, the audience is interested to know what is the cost of your proposed technique compared to traditional methods?
Yeah, that's a very good question. So yeah, indeed, for the traditional cost of generating DNA fragments, it's generally cost like $7 per DNA fragments. And then if we want to assembly them together, say if there are 6 or 8 fragments, so each plasmid may cost around $50. But by using our robotic system, indeed, if we only do several or like 10 or 20 plasmids, that's not worth to use the robotic system. But if we are building a large library such as 100 or 200 or building a lot of plasmids for the company, then this will save a lot of money. So the only cost we have is to getting the primers. Then the downstream parts, all the equipments are involved, and those are standardized processes for every kind of plasmids, and also other projects as well, not only for building plasmids.
I see. Okay. So there is another question. Is there a limit in the number of plasmids you can put together in a single reaction? And how do the robots deal with the differences in the sizes of the plasmids?
Yeah. So so the limitation of how many plasmids really depends on the plate we are using. So currently we are using a 96 well formatted plate. So ideally we can do 94 plasmids at the same time, because we want to leave 2 wells for checking, like putting the control, like positive controls and those things. But we can also always increase the plasmids in one pot to 384 or even 1536 wells. But yeah, as you may know, if we increase the amount of plasmids, there will be more chance to getting unexpected errors. So currently we are I would say we are limited, or we are focusing on 96 plasmids at the same time.
I see. Okay.
Yeah.
So you also mentioned that you need to ask a company to synthesize primers before generating plasmids. Were you thinking about automating it fully? That is, to start from automated order reading and primer synthesizing?
Yeah, that's a good idea. Like currently, yeah, we are generating the Excels and listed all the things we want to order. Then we send for certain companies such as Twist Bioscience or IDT to synthesize for us, and that will cost money. But in the future, like in universities or other like collaborators, we may start from the scratch just to synthesize the primers by ourselves. Yeah, but that will definitely be a future goal.
I see.
Go, yeah.
I see. All right. So thank you very much for sharing your presentation with us here today. I think that's all the questions we have.
Thank you. Thank you very much.
Thank you very much.